We are from Subgroup 4, STB1083. My name is Yosili, 106745.
My name is Tan Sing Ro, 106530.
My name is Aisha Nadira, 97234.
My name is Alicia Gabriel Jong, 103230.
So, in this practical session, we will do practical 1, which is spectrophotometry protein analysis. This experiment has 3 main objectives. So, the first one is to understand the principles of spectrophotometry for the qualitative and quantitative analysis for biological molecules. And the second one is to construct a standard calibration curve using bovine serum albumin BSA protein standards. And lastly, the third one, to determine the concentration of unknown protein samples A, B, and C by applying the Beer-Lambert's law and the standard curve.
These are the material and apparatus that we use in this experiment. The materials that we use are Bradford reagent, BSA protein with the concentration of 1 milligram per milliliters, unknown sample A, B, and C, and also distilled water. For the apparatus, we use micropipettes and their tips, test tube and test tube rod, spectrophotometers, cuvette and cuvette tray, and also laboratory mixer. In part 1, micropipetters was used to place 4 ml of distilled water into 7 test tubes. Next, 1 ml of Bradford reagent was added into each test tube. "50 microliter, 100 microliter, and 200 microliter of BSA protein standard was then added into the test tube in duplication. Each test tube was well mixed using laboratory mixer. Two ml of mixed solution were placed into the cuvettes. A sample without protein has prepared as blank to zero the machine. The sample read from the list concentration, and the adsorbance were recorded for each. Average and standard deviation for each point were calculated. Graph was plotted for the result, including the arrow bar. For part 2, repeat step 1 to 6 in part 1, but replacing the BSA protein standard with 3 unknown concentration sample A, B, and C. The adsorbance of each unknown protein concentration were then measured at 595 nanometer. The reading of each unknown protein concentration of A, B, and C were recorded. The concentration of the unknown protein sample was estimated using the standard curve plot.
Now I will be presenting the conclusion of the practical 1. In conclusion, these experiments show a clear relationship between protein concentration and absorbance. In part 1, absorbance increased consistently as the volume of BSA increased, while in part 2, a linear standard curve with an R-squared value of 1.0 was obtained. Using the equation y equals to 0.02x, the concentration of the unknown samples A, B, and C were determined at 5.60, 4.75, and 5.40 micrograms per milliliter respectively. These results are consistent with the Beer-Lambert's law, which shows that absorbance is directly proportional to the concentration. The strong linear relationship also indicates that the experimental results are reliable. Therefore, the objective of these experiments was successfully achieved.
So next, we will move on to our group reflection. So the first one will be our new learnings. So firstly, we learn on how the spectrophotometer works, which is by illuminating light through the clear part of the cuvette. The detector will measure the light intensity transmitted through the sample using blank sample. We also learn that blank is important in order to eliminate the background interference. Secondly, we learned the advanced laboratory technique, specifically on the micropipetting and also handling. We learned the precision use of micropipettes, which is P20, P200, and also P1000. We mastered the two-stop techniques, and we also learned that the clear sides of the cuvettes must remain untouched in order to prevent inaccurate readings of the absorbance. The third one, we learned on how to analyze data using a curve. We learned on how to find the unknown concentration using the linear equation derived from the graph. And lastly, we learned the importance of the accuracy and also precision. We learned that even a small error in the experiment can give a significant impact on the result.
Secondly, one of the main challenges we faced was maintaining accuracy and consistency throughout the experiment. Small variations in pipetting technique or sample preparation could lead to noticeable differences in absorbance values. We also realized that factors such as improper cleaning could interfere with light transmission and affect the readings. In addition, coordinating tasks within the group require effective communication, as any misstep in procedure could impact the overall results. Lastly, for improvements, we suggest increasing the number of replicates to enhance the reliability and reproducibility of the data. Assigning clearer roles within the group before starting the experiment could also improve workflow and reduce errors. Furthermore, taking extra care in instrument calibration and sample handling would help minimize systematic and random errors. Overall, this experiment not only strengthened our understanding of protein quantification using spectrophotometry, but also highlighted the importance of precision, teamwork, and critical thinking in obtaining accurate scientific results.